Hello, my name is Sarah Coulthurst, and I'm very happy to have the opportunity to tell you about some of the research that has been going on in my lab over the last 12 years. The first idea that I need to introduce is that, in the real world, bacteria do not live by themselves or in uniform populations made up of all the same bacteria. In fact, bacteria live in mixed communities containing many different types of bacteria and often also other microorganisms, such as microbial fungi. As illustrated on the left, mixed bacterial communities can be found almost everywhere, from soil and plant roots, through marine environments, to geological niches and, very importantly, associated with humans and all other kinds of vertebrates and invertebrates. Bacterial communities can be found all over the body, including on the skin, in the mouth, in the respiratory tract, and of course in the gut. These are normally harmless or even beneficial to our health. However, in some cases, harmful pathogenic bacteria can invade these communities or other sites in the body, causing infections and disease. Bacteria in these mixed communities are constantly interacting with each other. Sometimes these interactions are cooperative, for example, working together to gain nutrients or to protect the community. However, they're often competitive, as different bacterial species compete with each other to gain access to limited space and nutrients in a particular niche. This competition can be passive, simply trying to be better at acquiring a particular resource than your rivals. However, very often there is active competition between bacteria. The bacteria try to directly kill or disable their competitors. As shown in the middle panel, there are two main ways that active competition operates. The first is by the production and release of small molecules or proteins which can diffuse through the environment to reach and kill other bacterial cells, for example antibiotic molecules. The second is more sophisticated. The attacking bacteria physically contact the targeted cells and directly inject toxic proteins into them, usually killing them very effectively. These contact-dependent antibacterial systems are an example of an important type of weaponry that bacteria use to interact with their environments, namely protein secretion systems. As illustrated in the right-hand box, protein secretion systems are nanomachines that bacteria use to deliver specific proteins to their external environment or into neighbouring cells. There are different types of these machines, and they're used for many important processes, including injecting toxic or behaviour-modifying proteins into host plant or animal cells during infection, attacking rival bacteria, or releasing proteins to the environment which allow the bacteria to acquire nutrients or become attached to a surface. In my lab, we study one of the secretion systems that is used for active contact-dependent competition between bacteria, called the type 6 secretion system. The schematic on the left summarises what you need to remember about the type 6 system. It's a nanomachine that many different bacteria use to fire toxic proteins, called effectors, directly into neighbouring cells. The type 6 system is a very versatile system that can deliver effectors into eukaryotic cells, including host cells during infection, or to the environment. However, its primary and most widespread role is as a weapon during interbacterial competition. In this case, the secreting cell delivers antibacterial effector proteins into a rival bacterial cell, efficiently killing or disabling it. There is now accumulating evidence that type 6 systems play a key role in shaping the composition and dynamics of many microbial communities, including those important for health and disease. The type 6 system fires effectors into a target cell by a mechanism reminiscent of a spear gun or crossbow. As shown on the right, the system physically propels a rigid puncturing structure consisting of a long tube tipped with a sharp spike out of the secreting cell and into a neighbouring target cell. This puncturing structure is decorated with different toxic effector proteins inside the tube or on the outside of the spike, and these are carried into the target cell and released. The power for this propulsion event comes from a long contractile sheath which assembles around the tube inside the cell before the firing event. The sheath assembles in a high-energy extended state, like a stretched spring, and then rapidly contracts, propelling the puncturing structure with its loaded effectors out of the secreting cell and into the target cell. Following the firing event, the machinery is disassembled, ready for another round of firing. We have studied the role of the type 6 secretion system and how it works using a bacterium called Serratia marcescens as a model. Serratia marcescens is widely found in many different environmental communities, including in soil and water, and can be a highly virulent pathogen of insects. Importantly, it can also cause hospital-acquired infections in immunocompromised patients, and these are often associated with multi-drug antibiotic resistance. A large-scale genomics study and other work in our lab has revealed that all strains of Serratia marcescens carry the same basic type 6 system, which is encoded by a large cluster of genes. The Serratia marcescens type 6 secretion system has potent antibacterial activity against many different bacterial competitors. If we co-culture a particular strain of Serratia marcescens with other bacteria, in other words, if we mix them together in equal amounts and leave them to interact with each other for a few hours, we can see that the Serratia marcescens effectively kills the competitors. For example, only 3 in a million cells of E. coli survive being exposed to Serratia. Similarly, only 6 in 100,000 Pseudomonas cells survive, and only 2 in 10,000 cells of a different strain of Serratia marcescens, showing that the type 6 system can be used against even close relatives. We know that this killing is due to Serratia's type 6 system because if we inactivate it, all of the competitors survive. We've also learned that Serratia deploys its type 6 system according to an offensive strategy, firing preemptively rather than waiting to be attacked as some other bacteria do. This means that as well as aggressive competitors, Serratia can also effectively kill peaceful competitors and will still fire in the absence of close contact with other cells. So how does the Serratia marcescens type 6 secretion system manage to achieve such efficient killing of other bacteria? Like other antibacterial type 6 secretion systems, it does so by delivering multiple antibacterial effector proteins with different toxic activities. What all these effectors have in common is that they damage or destroy essential components of a bacterial cell. These are cellular components that are the same or very similar in all types of bacteria, and without them the bacterial cells die or cannot grow. So far, we've identified 8 antibacterial effectors in our model strain. As an example, I will first mention 2 of these, called SSP1 and SSP2, shown in green. The cell wall around the outside of bacteria is a rigid meshwork structure made of peptidoglycan, and it's required to maintain the shape and strength of the cell. SSP1 and SSP2 have peptidoglycan amidase activity, which means that they cut up the cell wall of target cells by cleaving the peptidoglycan at specific sites. When these effectors damage the cell wall, the intoxicated cells swell, elongate, and ultimately lyse, or burst open, as you can see from the pictures of the SSP2 intoxicated cells in the inset on the left. Of the other effectors, 2 of them, RHS1 and RHS2, destroy essential cellular molecules inside the cell, namely DNA and NAD. 2 others, SLP and SSP6, compromise the integrity of the inner membrane, which is the membrane surrounding the bacterial cytoplasm. The inner membrane must remain tightly sealed against the passage of ions in order to allow the cell to maintain its energization. SSP6 is a pore-forming effector which allows specific ions to flow through the inner membrane, causing de-energization of the cell, whilst SLP probably attacks the lipids making up the membrane. We're still working on the precise mode of action of 2 more effectors. One thing that may have occurred to you by now is, how does the secreting cell avoid intoxicating itself and its identical sibling cells with its own toxic effectors? The answer is that the secreting cells have specific matching protection proteins called immunity proteins for each of their effectors. These immunity proteins neutralize the toxicity of their partner effector by binding to it tightly to prevent its activity. In the example in the inset, which shows the effector SSP1, which is in green, bound to its matching immunity protein, which is purple, the immunity protein is binding and blocking the place on the effector which actually holds and cuts the peptidoglycan, known as the active site, and this prevents the effector from working. Immunity proteins are located in the part of the cell where their effector acts, for example in the same place as the peptidoglycan, so they're ready to intercept incoming effectors and prevent their toxicity. So we now understand quite well how the Serratia marcescens type 6 secretion system is used to kill bacterial competitors. However, it turns out that antibacterial effectors are not the whole story. We've used the type 6 secretion system of Serratia marcescens as a model to show that bacteria can also use the type 6 system to kill microbial fungi. This isn't really surprising because many microbial communities contain both bacterial and fungal species. If you look at the pictures on the left, you can see what healthy fungal cells look like in the no bacteria column. Saccharomyces cerevisiae is brewer's yeast, while Candida albicans colonises the human body and can cause disease. In the middle column, you can see that the fungal cells look sick and damaged when they are exposed to Serratia. The picture at the bottom shows dead cells stained in pink. But when the Serratia has an inactivated type 6 system, as on the right, the fungal cells are fine. In fact, when we co-culture Serratia with fungal cells, we find that less than 10 fungal cells per 1,000 survive. But again, there's full survival if the Serratia have an inactivated type 6 secretion system. Finally, if you look at the pictures on the right, you can see that Serratia cells attach closely to Candida cells, which will allow the contact-dependent effector delivery to occur. And you can also see in the rightmost picture some dying Candida cells following their exposure to the Serratia type 6 secretion system. We then went on to show that the Serratia marcescens type 6 system has antifungal activity because it delivers different effectors which specifically act against fungal cells. These two antifungal effectors are called TFE1 and TFE2. TFE1 causes a loss of membrane potential across the membrane around the outside of the fungal cell, which means that the cell loses its ability to transport molecules in and out of the cell properly. TFE2 intoxication leads to a disruption in nutrient uptake and metabolism, particularly of amino acids, which means that the cells start to enter some kind of starvation. We're currently working on trying to understand exactly how TFE1 and TFE2 cause these harmful effects. Putting together these antifungal effectors with all the antibacterial effectors I told you about earlier, we've learned that the Serratia type 6 system is a versatile and formidable antimicrobial weapon that acts against both rival bacteria and fungal competitors. When we identified and characterised TFE1 and TFE2, this was the first time that anyone had shown that dedicated antifungal type 6 effectors exist. However, we believe that many other antibacterial type 6 secretion systems in other bacterial species will also have antifungal activity. This would mean that the type 6 system is used to allow bacteria to attack both bacterial and fungal competitors in many different microbial communities and environmental niches. Why is all this important? We have an urgent need to find new drugs or strategies to combat infections caused by harmful bacteria and fungi, particularly as the problem of antimicrobial resistance continues to increase. As you've seen, bacteria can kill bacterial and fungal cells very effectively using the type 6 secretion system. So we hope that in the long term, we can learn lessons from how they do this and translate them into new tools in healthcare and agriculture. In summary, here are the messages I would like you to take away from our work. Bacteria can use the type 6 secretion system to effectively kill competitor bacteria by firing multiple different toxic effector proteins into them. The type 6 secretion system is a versatile antimicrobial weapon which can also kill fungal competitors, including pathogenic fungi. And we hope that understanding the type 6 secretion system and its effectors may contribute to new strategies to combat bacterial and fungal infections. Finally, and most importantly, I must thank and acknowledge all of the people who have contributed to this research, members of my lab, past and present, my scientific collaborators, and many other colleagues at the University of Dundee. I would also like to thank Wellcome and all the other funding bodies who have supported us.